papaya leaf, okra leaf, ampalaya leaf, tobacco, Erlenmeyer flask, dropper, glass slides, tweezers, diluted HCl, microscope, and measuring tape.
So first we'll label everything, from the materials and also our specimens. It is important to do this for us to work easily and efficiently and to avoid confusion.
Tobacco dried tea breeds, microscope power 10 without HCl. The body of dry adulterant named tobacco, which is dry under the microscope, as it resembles the color the lava coming out from volcano. On the other hand, the edge part of adulterant, it has cystolytic hair at the edge, and the bend of the lip looks like the nerves that we can see at someone's eyeballs.
Dried tobacco leaf with diluted HCl. We place the dried tobacco leaf with diluted HCl under the microscope, and as we observe, it has a lot of circle shapes that looks like a bubbles. The outside part of the bubbles has a hair that looks like a fiber of cotton, and the color of the dried tobacco leaf is red-orange. Subsequently, the edge part of the dried tobacco leaf, the cystolytic hair appeared more, and based on what Mr. Arceliano said, that it looks like the filter of a cigarette. Ampalaya without diluted HCl. The drug adulterant ampalaya leaf or the bitter gourd without diluted HCl, when placed under the microscope, we observed that the body of the ampalaya leaf, which shows the color green of the leaf, was more vibrant when seen under the microscope. The lamina leaf of the ampalaya has a circle shape like the egg of a frog. The midrib of the leaf shows the veins of the leaf that is similar to a road that we can see on the Google map, and the lamina leaf is like a styrofoam combined with glitters. On the other hand, the edge part of the ampalaya shows the cystolytic hair. It's like the skin of the cactus, and the ampalaya leaf looks like the San Francisco leaf. The lamina leaf of the adulterant shows a green color, and the parenchyma cells looks like the scale of a fish. Ampalaya with diluted HCl. As we placed under the microscope the specimen of ampalaya leaf with diluted HCl, and as we observed, there was nothing changed except the appearance of the bubble, and maybe it is due to the diluted HCl. On the other hand, as we move to the edge part of the specimen, the cystolytic hair does appear, and also the edge part of the specimen has a watery substance, and as we guessed, it is due to the diluted HCl that we placed.
The midrib looks like a road filled with snow. The vein that surrounds the leaf looks like a Google map. The veins are color white. The papaya leaf, when under the microscope, has plain and soft textures. As we perceive, on the edge part, the papaya leaf has no cystolytic hair. The vein thus appeared, and also it resembles the San Francisco plant. As we place the papaya with diluted HCl on the microscope, there was no changes except that we have seen bubbles and we had guessed that it was due to the diluted HCl. On the other hand, age still was no changes of the specimen.
The body of okra leaf, when under the microscope, has veins, and also the veins are color white, which really appeared during our observation. While the edge, there was no changes, except that we had seen a cystolytic hair in the laminar leaf of the okra.
The body of okra leaf, when under the microscope, has bubbles, and the veins is color white, which really appeared during our observation. While the edge, there was no changes except that we had seen a cystolytic hair in the lamina leaf of the okra. These specific characteristics mentioned below are based on information from various internet sources, including McGill University, Grow with EC, YouTube, Sensei Seeds, Snopes, Royal Queen Seeds, and Witty Optics. These sources contribute to our understanding of the microscopic features of dried cannabis leaves. When dried cannabis leaves are observed under a microscope, several characteristics can be observed. First is the trichomes. Trichomes, these are tiny crystal-like structures that cover the surface of cannabis leaves. They contain cannabinoids and terpenes which contribute to the plant's therapeutic and psychoactive properties. Under the microscope, trichomes can appear as mushroom-like structures or tiny hairs. Second is stomata. Stomata, these are small openings on the surface of cannabis leaves that allow for gas exchange. They can be observed under the microscope, and their presence and distribution provide insights into the leaf's psychological functions. The body of the marijuana leaf exhibits a captivating visual appearance when observed under a microscope. It can appear as if a river of lava is flowing with numerous circular shapes resembling bubbles. The texture of the leaf resembles the back of a turtle with its intricate and textured surface. The color of the leaf can resemble rusted metal, adding a touch of rustic charm to its overall aesthetic. Moving to the edge of the marijuana leaf, they are adorned with abundant cystolytic hairs. These hairs give the leaf a textured appearance similar to the scales of a fish. They contribute to the overall beauty and uniqueness of the leaf's structure. Microscopic examination allows for a closer look at the cellular structure of dried cannabis leaves. The arrangement of cells, veins, and other plant structures can be observed, providing insights into the leaf's composition. The pigmentation of dried cannabis leaves can also be observed under the microscope. Different strains may exhibit a variation in leaf color, ranging the vibrant green to purple or even reddish hues. It's important to note that the specific characteristics mentioned are based on information from various internet sources, including McGill University, Grow with EC, YouTube, Sensei Seeds, Snopes, Royal Queen Seeds, and Witty Optics. These sources contribute to our understanding of the microscopic features of dried cannabis leaves. These specific observations mentioned below are based on information from various internet sources, including McGill University, YouTube, Witty Optics, NCBI, and Snopes. These sources contribute to our understanding of the microscopic features of cannabis leaves. When cannabis leaves are diluted and observed under a microscope, several interesting observations can be made. Even when the leaves are diluted, the tiny crystal-like structures known as trichomes can still be observed. These trichomes contain cannabinoids and terpenes. which are important components contributing to the plant's properties. The body of the marijuana leaf, known as the lamina, exhibits a combination of red, orange, and yellow colors. This vibrant coloration adds to the visual appeal of the leaf. On the edge of the leaf, the presence of cystolytic hairs can be observed. These hairs give the leaf a textured appearance, resembling the scales of a fish. They contribute to the overall structure and aesthetics of the leaf. There may be the presence of a watery substance which could be a result of using diluted hydrochloric acid or the HCl during the observation process. However, it's important to exercise caution when using chemicals for microscopic examination. Diluted cannabis leaves still reveal the cellular structure of the leaf by observing the arrangement of cells, veins, and other structures. Valuable insights can be gained regarding the leaf's composition. The small openings on the leaf surface, known as stomata, may still be visible under the microscope. These openings play a crucial role in gas exchange and provide information about the leaf's psychological functions. Even when diluted, the pigmentation of cannabis leaves can still be observed. Different strains may exhibit variations in leaf color, which can be seen under the microscope. It's important to note that the specific observations mentioned are based on information from various internet sources, including McGill University, YouTube, Witty Optics, NCBI, and Snopes. These sources can contribute to our understanding of the microscopic features of cannabis leaves.
After conducting microscopic examinations of okra leaf, bitter gourd, papaya, dried tobacco, and dried marijuana leaf, it becomes evident that each specimen possesses unique anatomical characteristics. Okra leaf displays distinctive trichomes and epidermal cells, while bitter gourd exhibits a complex arrangement stomata. Papaya leaf showcases unique parenchyma cells and leaf veins. Dried tobacco leaf presents characteristic glandular trichomes and stomata patterns. Finally, dried marijuana leaf is distinguished by its glandular trichomes and irregularly shaped epidermal cells. These observations underscore the importance of microscopic analysis in botanical identification and highlight the diverse structural adaptations of plant species for various ecological functions. In conclusion, microscopic examination serves as a valuable tool in identifying drug adulterants such as papaya, okra, bitter gourd, dried tobacco, and dried marijuana leaves. Through careful observation and analysis, characteristics features unique to each substance can be identified, aiding in the detection and prevention of adulterant in pharmaceutical and recreational products.